Creatine is a non-protein amino acid that is synthesized mainly in the liver, the kidneys, and the pancreas. It's in the liver, the kidneys, and the pancreas where the transamination of amino acids, methionine, arginine, and glycine. Transamination meaning the transfer of an amino group from one molecule to another make creatine, which once synthesized is sent through the bloodstream primarily to skeletal muscle, but also to other tissues with high energy demands, such as the brain. The human body synthesizes approximately 1 gram of creatine per day. So, to have higher concentrations of creatine, we can look to our diet where we can consume animal products that have approximately 1 to 2 grams of creatine per pound. However, it's at 3 to 5 grams of creatine ingested per day that we start to see the benefits of creatine ingestion. So the easiest way to get that high concentration of creatine is through creatine supplementation. 95% of creatine is stored in our skeletal muscles, which have the role of voluntary movement and physical activities. Creatine by itself doesn't actually have much of a function for the skeletal muscles, but with the help of an enzyme called creatine kinase, creatine turns into phosphocreatine, where it is used as an energy reservoir and is called upon during high intensity exercises such as sprints, bench presses, and squats. Reservoirs of phosphocreatine turn into ATP, which is a molecule made in the mitochondria of human cells, and it provides our cells with the energy to carry out their various functions. Energy is released when ATP is broken down into ADP, or adenosine diphosphate, and a phosphate, and that energy is used to carry out the function of the cell. When our body performs a high-intensity physical endeavor like a squat, bench press, or sprint, the muscles that contract in that given exercise need immediate energy. However, the production of ATP through the mitochondria is not able to get to the contracting muscles quick enough. To get energy very quickly for high-intensity activities, our body uses the ATP-PCR system, or the PCR shuttle. The ATP produced in the mitochondria from the oxidative phosphorylation system encounters the mitochondrial creatine kinase. The mitochondrial creatine kinase will then allow for the creatine to become phosphocreatine by taking the leftover phosphate from the ATP that was broken down into ADP and a phosphate. The phosphocreatine created can then get through the mitochondrial membrane faster than ATP into the cytoplasm or sarcoplasm of the cell, where it will encounter creatine kinase again. Because of there being a lot of ADP available in the cytoplasm or sarcoplasm of the cell during exercise, the phosphocreatine is easily able to transfer its phosphate over to the ADP to make ATP for the cell to use. The ATP will then be used by the muscle cells to contract, and the creatine will return to the mitochondria, where this process will then be repeated.